Hello. So I'm restarting. I'm explaining the same concept of vitamin C. This vitamin C acts as a, it is not exactly called as a coenzyme, but it works and helps the enzyme to function. Now let me just get the feedback from you. Hello. Okay. So I'll continue. So here we are discussing the concept of vitamin C, how vitamin C works. Very quickly, I will elaborate again that these vitamin C, they act as a specific antioxidant, and during this process, what also they do is that they help in synthesize the collagen. During the process of collagen synthesis, this enzyme prolyl hydroxylase has to be regenerated, and during the process of regeneration, the help comes from ascorbic acid. So here, if the ascorbic acid is not present, your collagen, which is a triple helix, it and forms an interstrand hydrogen bond is not being synthesized. Now, today I'm going to talk about different vitamins which come under the class water soluble vitamins. And these vitamins are thiamine, which is known as vitamin B1. My major concern is not with the third, is basically for this column, third column, which is showing what kind of enzymatic reaction they can help. So actually what will happen, if I look at the vitamin alone doesn't work. Vitamin has to first convert it into the coenzyme, and once it is converted into coenzyme, so this has to be converted into coenzyme. So suppose there is a process which blocks the conversion of vitamin into coenzyme, the reaction will not occur. So what you have is that when you have this vitamin B1, they will be first converted into thiamine pyrophosphate. So this is the one which is coenzyme. Now what it will do is that it will react with the enzyme, and then once it binds with the enzyme, because it is a coenzyme, what it will do is that it will do aldehyde transfer. So actual reaction is a aldehyde transfer. So which enzyme they will bind? They will bind with the transferase. Then again, there is a vitamin B2, which is known as a riboflavin. It is the one, the coenzyme form is a flavin adenine dinucleotide. So if you look at thymine or if you look at flavin adenine dinucleotide, what you are looking at is that they are modified. For example, here pyrophosphate group has been attached. Here, if you see, if I take riboflavin and ATP, you will have adenine dinucleotide which has been attached. So actually the coenzyme is being synthesized from the vitamin. So major, one of the base residue is coming from the vitamin, and the rest part is coming either from phosphorylation reaction. So somewhere energy is involved when vitamin is converted into coenzyme. Same way, B6 is converted into pyridoxal phosphate. So you have pyridoxine converted into pyridoxal phosphate. So now these are these steps. Your vitamin will be converted into coenzyme, will be binding with the enzyme, and the enzyme will catalyze this typical reaction. For example, here transferase, here oxidoreductase. So these enzymes, they are helping. Now, with this help, what can they do is that they can take care of deficiency of the vitamin. For example, if thiamine is absent, you will get beriberi, where weight loss, heart problems, these are occurring. These are the last one is basically for medical doctor to understand how this vitamin deficiency is helping. But what we are trying to help you is that understand that the vitamin, once converted into coenzyme, they become part of the enzymatic reaction, and that's the way the catalysis occurs. Now, third one I have talked about is pyridoxine forming pyridoxal phosphate, and it is involved in the group transfer to or from the amino acid. So you'll be looking at that there are certain groups which are transferred from the amino acid to other group and other group to the amino acids. Then another example is nicotinic acid or niacin. It is converted into NAD plus. You will see that this is very, very important. When it is reduced, it can take the electrons, and they are the one which are important for biological oxidation reduction reactions. So when we talk about the reactions which are happening in the biological oxidation process or in the process of energy generation, we'll see that this NADH is playing an important role. In another set of reaction, you will see that not NADH, but NADPH is playing a role. So we'll talk about how the NADH is different than NADPH and how these two can play a very different role. Even they can give you a signal. Suppose instead of NAD plus, if NADP plus is required by the enzyme, that means the energy is very high. That is why NAD is phosphorylated, and once NAD is phosphorylated, it will carry out the reaction, which will be more like synthetic reaction rather than energy producing reaction. So there is a one type of reaction occurring in the body is for energy production, another is for the synthesis of important biological molecule or macromolecule. Next, vitamin is a pantothenic acid. It is converted into coenzyme A, and you will see that coenzyme A is a very important enzyme for acyl group transfer. So it will be involved in acylation reaction. Of course, it will be leading to its absence is leading to hypertension, etc. Then biotin, it's a very interesting enzyme. It forms a complex with a lysine. Lysine is coming from an enzyme. So your biotin and lysine forms a complex which is known as a biocytin. And this biocytin is a coenzyme. So this is the one which is responsible for carrying out or helping the enzyme. And what enzyme they will help is the one which is involved in the carboxyl group transfer. So you see that each coenzyme has a very specific niche enzyme with which they are going to work, and they are going to catalyze a reaction. Many of you might have learned about B12, and B12 is a cobalamin, and this is the one which is responsible for transfer of methyl groups. And of course, they are also in other reaction where they are involved. But very important thing is that they call anemia or pernicious anemia when they are absent. So you have on one hand the vitamin, they are converted into coenzyme. Once they are in the coenzymic form, the concept is that they will be acting along with the enzyme. So if I say that just thymine pyrophosphate is present, will they catalyze the reaction? Answer is no. If I have just vitamin, will they catalyze the reaction? No. So first you have to have vitamin. Vitamin should be converted into coenzyme, and then you should have an enzyme as well. So once you have this coenzymic formation plus enzyme, these reactions will be catalyzed. Though I have covered it in one PPT, there is a lot of reaction mechanism, etc., can be understood, but I think at this point of time you do not require. Actually, if I go by real chemical process, these coenzymes, they are helping in the enzyme catalysis either by in some places by transferring the substrate molecule from one part to another part, in some places taking electrons, in some places giving electron. So that is where they are responsible for actual chemical process. Now coming to the fat soluble vitamins. Now fat soluble vitamins, they are actually not have been assigned to any type of coenzymic function, but still they are responsible for certain important reaction, and that's why we are going to talk about. So when we talk about this fat soluble vitamin, they are A, D, E, K. So these four vitamins, they are fat soluble. Do we have vitamin C also fat soluble? Answer is no. Vitamin C is the one which is given by a letter code, but still it is a water soluble vitamin. So coming to one of the fat soluble vitamin, vitamin K, the term K has come from coagulation. Coagulation is a German term which means actually blood coagulation we are talking about. So it is because if you vitamin K deficiency is occurring, then under those conditions, blood clotting is impaired. And that is why this vitamin K is required for coagulation. What it does is that it participates in carboxylation of glutamate residue to alpha carboxyglutamate. So now, do you recall we have discussed that the proteins, once synthesized, its amino acid can be covalently modified. So there we have discussed about the concept that how vitamin K can modify glutamate into our alpha carboxyglutamate. You have some of you have written this answer also in the last minor. So it makes the modified glutamic acid, which as a much stronger chelator of calcium ion. So actually, it chelates calcium ion, and that's why they proceeds for the coagulation of the blood. Another very important vitamin, which is a fat soluble vitamin, is vitamin A. So what vitamin A does? Vitamin A is a chemical, is a retinol. It is formed from carotene. So if you have a carotene, all of you know that if we do not eat proper vitamin containing vegetables like carrots, like radish, you have a deficiency of vitamin A. What it means is that vitamin E is derived from beta carotene, and if you break the beta carotene from center, you get two molecules of vitamin A. Here I have written retinol. It is the chemical which has got hydroxyl group. Vitamin A can also be in the form of retinal, Al, that means it will be aldehydic form. Somewhere you can also find retinoic acid, that is a acidic form. So you have the same molecule, but the terminal is modified. It could be alcoholic, it could be aldehydic, it could be acidic. Now, precursor of retinal, the highly sensitive group present in rhodopsin and other visual pigment. So if you look at the vision, how our vision works, it works with a protein which is known as a rhodopsin. Rhodopsin undergoes, changes, and here vitamin A plays a very important role. Now, if you will, dig up into the detail, why should vitamin A work? When you will look at the vitamin A structure, you will find that it has a conjugated double bond. And through this double bond, your electron transfers, so our photons can transfer. And that's the way they are acting as a very good photon acceptor. So what you have is you have a light sensitive group in rhodopsin, and this rhodopsin binds with a vitamin A, and that's the way it carries out its function. So a deficiency of this vitamin A leads to night blindness, and in addition, it is involved in other things like vitamin A is in growth factor, or it can also, retinoic acid can also contain a terminal carboxylate, which is interesting for, sorry, I have to go back, and it is involved in the gene regulation, etc. So these are some of the important functions of vitamin A. Let us move on to vitamin D. It is a metabolite of vitamin D is a hormone that regulates the metabolism of calcium and phosphorus, as well as the vitamin D deficiency. Because it is regulating the hormones, its deficiency can cause bone formation in growing animal. Uh, Now, vitamin E, many times its function is not very well known in a higher organism, but it is involved with the infertility in rats because it's also known as a chemical name of vitamin E is alpha tocopherol. The vitamin also reacts with and neutralizes reactive oxygen species such as a hydroxyl group, radical, etc. And that is why it is understood that it is also involved in protecting our membrane lipids, and that's why in damaging cell structure. So that is why what you find is that many these this, many things they contain vitamin E where you require the protection from the oxidation process. So that is where vitamin E has been used. So if I look at, in summary, what fat soluble vitamin does is that it plays a role in vision, growth, and reproduction, which is vitamin A. vitamin D in regulation of calcium and phosphate metabolism, vitamin E acts as an antioxidant, then vitamin K is involved in the blood coagulation. The deficiency, of course, vitamin D deficiency, very common in children, that causes rickets, whereas vitamin A causes night blindness. I'm just highlighting the most important thing. And in case of vitamin E, it acts as an antioxidant. And in K, if it is absent, the coagulation is inhibited. Now, for today's class and the previous class, let us summarize it that enzymes you have learned they are very powerful and highly specific catalyst. Now, do you know that why the catalytic power comes? How the catalytic power comes? Catalytic power comes because your enzyme binds with the substrate and it has got certain structure where the binding can occur. So the specific sites which are present in the structure of enzyme is the primary reason why the enzyme has a high catalytic power and is highly specific. And it is more important because they are made up of proteins. Proteins are made up of chiral amino acid, and these chiral amino acid can have stereospecificity. The binding is the principal reason from where this binding energy is leading to the lowering of activation energy, and that's why enzyme works. Then free energy is a useful thermodynamic function for understanding the enzyme. So you have understood how thermodynamics is involved in the enzyme catalyzed reaction, and then enzyme accelerate the reactions by facilitating the formation of transition state. This concept we have discussed today, and it's very important that it is not actually the substrate, but it is this transition state analog of the substrate which binds with the enzyme and catalyzes the reaction. You have learned about the Michaelis Menten model, and this Michaelis Menten model accounts for the kinetic property of many enzymes, but some of them they may not follow. And basically, the enzymes which do not follow the Michaelis Menten model, they are the one which are regulatory enzymes. Now, enzymes can be inhibited by specific molecules, and these molecules are the one which may have the similarity with the structure of the active site. They may not have is a similarity. I have not given you details of enzyme inhibition kinetics. In your classes, when you study about the enzyme science and engineering, you will understand how inhibitors of enzyme they work, how they are important in different in different applications, and there are different types of enzyme inhibitor. One can bind with the active site, can inhibit, so they will be actually the competitive inhibitor. There may be non-competitive inhibitor which may not bind actually to actual site, but they can bind at other site. So you have a competitive inhibitor, non-competitive inhibitor, you can also have uncompetitive inhibitor. So there are different types of inhibitors of enzyme. They are present, their kinetics will be different. How they will be different? Because if something is a binding with the active site, what they can influence? They can influence the binding affinity, that is a Km. So those way I have not covered that portion, but yes, you understand that enzymes they can be inhibited by the various molecules. Then we have talked about the importance of vitamins, how the vitamins plays an important role where it can work as coenzyme after modification, and this coenzyme modification occurs mostly by inserting certain molecules in the vitamin structure. So these molecules are normally in presence of ATP molecule, they are converted into the coenzyme, and once coenzyme as well as enzyme. So enzyme is the protein portion, coenzyme is the organic part. And this, when interacts, they become functional enzyme and they show their reaction. So with this, I will stop. If you have any question, let me know. You can ask question. If I can't hear you, you can just write in chat and let us discuss. So do you have any question?
So I don't think there are any questions right now.
Okay, then I will sign off.